Okay, Professor, so I will leave the mic to you. I just want to say welcome everybody to the University of Kent weekly webinar, and I would like to have the honor to give the mic to Professor Darren Griffin. He is the director of the Master's Degree in Reproductive Medicine and IVF at the University of Kent in the United Kingdom, and also the director of the PhD program in the Reproductive Genetics and IVF at the University of Kent. So if you're looking for your PhD, this is where you start. And I will leave Professor Darren. He will introduce the faculty. There's Dr. Giuseppe here, and Mayyas, and of course the speakers. All to you, Professor. Thank you.
No problem. Right, we're coming up nearly up to time. So I believe you're going first. So I will ask you if you don't mind sharing your screen so it's ready for straight up a 4 o'clock start, if that's okay. So welcome everybody to the University of Kent seminar sessions in collaboration with the IVF Company and Tri-C Academy. It is a delight to welcome you all. I see that we have 73 attendees already. It is wonderful to see you, and please do fill out some questions in the question box and we'll do our best to answer them. It gives me great pleasure in the first instance to introduce all the way from the Yasmin IVF and Fertility Center, IFSL, who is going to talk to us about the revolutionary technique of non-invasive pre-implantation genetic testing. So over to you, Aya.
Okay, hello everybody. This is Aya Faisal, embryologist and the geneticist in Yasmin IVF and Fertility Center. I'm going to talk about a very interesting topic. I find it this way. It's the non-invasive pre-implantation genetic testing, and I think it's one of the promising techniques in the field of IVF. So here is my content, but I'm so excited to share the information I have in my presentation. So I'll jump to what's PGT. Well, PGT or the pre-implantation genetic testing, it's a set of techniques that aim for testing the genetic material of embryos in the IVF treatment before transferring them to the mother's womb, to check if there is any chromosomal abnormalities or any genetic disorders before transferring them. And this technique was initially introduced to the field of IVF by Dr. Handyside in 1989, and proudly saying that it was also initiated and done by Dr. Darren. And I'm so proud to mention that I'm one of his students. So basically, in what cases do we do PGT? Well, there are some cases of the advanced maternal age or the recurrent miscarriage in the repeated pre-implantation, the repeated implantation failures, and in previous pregnancies for aneuploid embryos, and also in severe male factors, and so many other things. Like in Egypt, some couples just request doing it to make sure that they have healthy babies. And speaking about Egypt, I would like to just have an insight about the decline in the fertility rate in Egypt. As you see here, it's dramatically increasing. As you see here in 2014, it was 3.5 and it dropped to 3.1, and that's according to the latest statistical analysis done by the Central Agency for Public Mobilization and Statistics in Egypt, which was done in 2019. So it means that we are in the need for a lot of embryologists out there. Also, I would like to mention why there is an increased demand on PGT in Egypt, because of the relatives marriage, which is somehow very common here in Egypt. It's the consanguinity, and also because of the advanced marriage age. And some people usually do it for selecting the sex of the embryo to avoid some X-linked or Y-linked disorders. So transferring healthy babies sounds perfect, like, you know, yeah, having disease-free babies, it sounds perfect. But everybody knows that it has some challenges because we subject the embryos to sort of damage. And also because of the mosaicism level, which would give us sometimes false positive or false negative results. And as everybody here knows, might know that most of the embryos are mosaic. And also one of the challenges is that we need highly skilled embryologists that would be able to take biopsies from the embryo without causing damage to them. So let's look at the history of taking biopsies. It started in 1987 by taking polar body biopsies, and it's still going on. And then it was and people started to take biopsies from the cleavage stage embryos, and then the plastocyst stage, and then recently the plastocentesis, which depends on the plastocyst fluid inside the embryos. And what I would like to highlight the most here now is taking the biopsy, or actually taking the DNA sample from the stent culture media, and it was introduced recently. So if I'm about to classify the techniques of taking biopsies, I would classify them into invasive and less invasive and the non-invasive techniques. So in the invasive technique, we somehow subject the embryos to damage. And also it affects the success of the IVF cycle, more or less. But what I found most promising here is the non-invasive technique. Not even the less invasive technique, because the embryo will still be subjected to kind of damage. So let's talk about the stent culture media for the embryos. Well, they found recently that there are some cell-free DNA that present in the surrounding of the media where the embryos are cultured. It was found that it even has more DNA than what's found the one that's found inside the plastocyst fluid. To be honest, the mechanism itself is still not completely clear. Another study also, and it's a recent one, that also found that the more we culture embryo, the more we found more DNA in the surrounding media, which proves that the DNA found is of an embryonic origin, and that's a very important point. We will go back to them. And yeah, there are like a lot of studies that has been working or actually trying to investigate this technique. And some successful trials was done for checking the TSPY gene and the TBC1D3 gene. That they were able to detect it by just taking the stent culture media to avoid the Y-linked disorders. It was only verified in 5 cases so far, but it's still a good study. So what's the mechanism? As I mentioned, it's still unclear, and that's actually one of the drawbacks, because we're still not sure if the DNA found is of an embryonic origin or not. But we can roughly say that it's because of the zona pellucida permeability that allows the transfer of the cell-free DNA from the inside the plastocyst in the plastocyst fluid to the surrounding media. So what's the procedure? Well, I'm gonna just sum it up in 5 steps, which will start by embryo culturing, span culture media collection, and then the whole genome amplification, then sequencing, and then analyzing the data we get. So on day 3, after we evaluate the embryos and choose the one of the best grades, we transfer them to fresh media and start culturing them from day 3 to day 5. Then we collect the spent culture media, and definitely we should also transfer the embryos to another media or cryopreserve them. Then they are subjected to whole genome amplification using the Malbec technique, which is a multiple annealing technique that's used to amplify the quantity of the DNA we have. And it's called the multiple annealing and looping based amplification technique. Then DNA sequencing using next generation sequence. And then the data we get is analyzed by a software specialized to sequence or to analyze the data we get. And the Chromo, which is developed by Yikon Genomics, could detect any deletions and also or duplications or whatsoever, and also could detect could be sensitive to the mosaicism level. So I would like to introduce the first successful Brazilian attempt to test an embryo using non-invasive PGT, and it was carried out to a Brazilian couple of 37-year-old woman and 36 husband. They were suffering from a secondary infertility, and both of them present normal karyotype. I'm not going to go through the medical investigation a lot, but I would say that they were normal and they were ready for the IVF treatment. We got the fertilization result was about 11 M2 oocytes were collected. 8 of them were fertilized. 3 embryos only reached the blastocyst stage, so they cryopreserved 2 and transferred 1 embryo. But unfortunately, after 14 days of waiting, the pregnancy test showed negative results. So that's why they decided to go and transfer the frozen embryos that were subjected to the non-invasive PGT. And here were the results. The first blastocyst actually showed that it was a female, as we see here. And the second blastocyst showed that he was a male, but he also had a sort of deletion in chromosome number 15. And I'm just gonna give like a hint about what does it mean, because actually the partial monosomy or a mosaic partial monosomy or deletion chromosome number 5 is usually associated by a syndrome called the Prader-Willi syndrome or Angelman syndrome, and it's well known for mental retardation or actually mental development delay. And yeah, luckily the blastocyst number 1 was euploid, so it was normal. So after transferring the 2 blastocysts, the beta HCG level was actually high enough to say that the woman was pregnant. They developed till week 7, but unfortunately one of them had an arrested development, and what continued was the embryo, the female embryo, because you see we're strong enough. So the woman gave birth to a healthy girl. So as you see, that was the successful attempt that took place in Brazil. And there are also recent studies that proves the potentiality of the non-invasive technique to detect monogenic disorders and the chromosomal abnormalities. And there was a recent study that showed that it could be efficient to detect the X-linked or the sex-linked disorders. Another recent study also tried to compare the non-invasive technique with the ordinary or the conventional invasive technique, and surprisingly, the non-invasive technique showed less significant false positive results than the ordinary PGT technique. Another study also tried to compare both of them together with the whole embryo, and surprisingly, the non-invasive PGT technique showed positive protective values of 93.5% and false positive rate of 6.5%, while the ordinary PGT showed only 78.4% and false positive rate of 21.6%. You see? So I can sum up the outcome of the non-invasive PGT if it was proven to be successful technique, that it will help us to get better results, less damage to the embryo, because we're not going to take any biopsies from it or subject it to any invasion or micromanipulation. We will also need less highly skilled embryologists, so we won't rely on the technicality of the embryologist much, and definitely it will be cheaper. And what's more important is that it will help us to get better IVF outcomes. But yeah, as anything in life that has pros, it also has some concern and cons. And yeah, a lot of concerns about the non-invasive PGT technique, because it's not only a new technique, but we still have a lot of unanswered questions. Like the DNA cross-contamination. We might find DNAs that they are not originated from the embryo itself. They might come from maternal cells, paternal cells, or even exogenous source like the embryologist or the lab personnel, or even the media itself. And we still also uncertain about the origin of the cell-free DNA. We have no idea about the mechanism so far. Actually, some people say that, as I said, it might become of a cross-contamination, and some say that they are coming from apoptotic cells, which will not be able to reflect the DNA of the embryo itself. We might also have insufficient quantity and the quality of the DNA that might also lead to false positive or false negative results in the results. So here's the question: PGT or non-invasive PGT? And now we gotta have a poll. I want you to give me your input, whether you are with PGT or the non-invasive PGT technique.
So we'd like you to split one of the following. Where do you think we're going to go, for PGT or for non-invasive PGT? So please do select one of the options, and the poll is in progress now. So you can see that it's filling up quite quickly, and we've got about 52% have voted right now. I will give it a few more seconds. And we are going to close the poll in now. There we go. The poll is closed. And I can say that we had a 60-40 split, Aya, and 60% said non-invasive. So there you go. Okay.
It's quite interesting because having a disease-free babies and not also affecting the implantation success or the success rate of the IVF treatment itself sounds perfect, yeah? But I think there are a lot of things that we have to consider before actually decide whether to keep relying on the PGT or to start doing more research about the non-invasive PGT and at least give it a try. Like, for example, we need to work on eradication of the DNA contamination or the DNA cross-contamination, and also we need to enhance the procedure itself. We need to decide on the best technique to collect the sample, and also we have definitely need to optimize the data, the data analysis software that we will rely on to get the results, and so many other things. But the aim of this presentation actually is, I wanted to report the first occurrence of the childbirth in Brazil that carried out the non-invasive PGT. And I will also take the chance to invite every embryologist out there in any country to start working on this, on enhancing or improving this technique, or at least give it a try and start reporting many new cases. As we here in Egypt, we are actually working on that under the supervision of Dr. Assam Khalifa, his the manager of the Yasmin IVF Center. And yeah, I would like to have a lot of research on that to decide whether if it could be actually a reliable technique that we could rely on, and we might have it one day as a routine test before transferring any embryo. So yeah, we are still under the experimental phase, and maybe if at least I think this is what I believe in, even if the non-invasive PGT technique, even if it failed to prove itself to be a promising technique, but I'm sure that it will help us to know a lot about the development or the nature of the embryo development and the genetic aspect of it that will definitely improve the IVF outcomes, and I'm sure that it will take the IVF to a whole new level. Thank you so much for listening to me. Here are my preferences, and you're more than welcome to have or to ask me any question about this presentation. Thank you so much.
Thank you very much for that presentation. It was fantastic. I'm going to start with a question, if I may. So you're based in Cairo. What's your link with Brazil?
Well, actually, it was the first reported case that of a childbirth that was carried out using the non-invasive PGT technique, and that's a recent one. It was reported, I think, yeah, last November or last March in 2020, and yeah, I really wanted to have it, like, you know, to encourage people out there to start reporting their first cases in many different countries, so that we can decide whether it could be a promising technique or not yet.
Okay, so we've got a couple of questions here. More or less the same question from Ekaterina and from Said, and it's to do with the accuracy. So when you compare, let's say, the diagnosis from the non-invasive with what with the actual embryo itself, follow-up, what is the accuracy rate in general terms?
As I mentioned in the presentation, it was around 93% that goes for the non-invasive PGT technique, that that was like a positive predictive value compared to the non-invasive, actually the ordinary PGT technique. I could share with you the paper.
Yeah. Okay. So this is this is one thing I couldn't quite work out. Is this comparing the concordance of the biopsy with the non-invasive? Has anyone done the whole embryo? Okay, got it. That's the thing I was most interested with. As I understand it, the aneuploidy rates are higher in the non-invasive compared to the trophectoderm biopsy. Is that still true, or?
Yes.
Mm-hmm.
Yes.
So you do get a greater aneuploidy rate. Okay, cool, excellent. Now, I just wondering if you had any comment on, there was a case in Australia recently where allegedly it was some sort of misdiagnosis, and there was quite a lawsuit that seems to have put the whole area of niPGT back a little bit. What's your opinion on that?
Sorry, I didn't hear the question properly. Could you please?
Yeah, in Australia, there's been a legal case where there was a allegedly a misdiagnosis of following non-invasive PGT, and the criticism was, the whole test hasn't been properly validated and it shouldn't be, put out for patients just yet. So I was wondering what your opinion on that was.
Yeah, I think we will find so many cases that reports, or actually that have the same opinion about the non-invasive PGT, because speaking about myself, I'm actually working on developing this technique to be able to decide on it. So yeah, one of the drawbacks of it, that because the quantity of DNA is not sufficient, so sometimes the amplification, and actually I don't want to go so much into technical stuff, but when they are subjected to amplification, it leads, because in a way or another, it leads to false positive results. And that's why I said we need to work on optimizing the technique of the whole genome amplification, and actually the next generation sequencing, and definitely the software, because they are not optimized yet to detect or actually to analyze the data derived from using the non-invasive PGT. So I'm sure that we will get a lot of with and against opinions, and actually the research and the more cases reports will be able to decide on that. That's from my opinion.
Excellent. And I guess this is Monash, and a number of one of our listeners, Suhar, is saying that there's been hundreds of wrong diagnoses at the moment. So I get the impression that it seems to be variable from center to center. So in Valencia, for instance, it seems to be very accurate, but you're seeing in other centers a much less tight concordance rate. And I wonder whether you had an opinion on how the things can be different from lab to lab, for what reasons?
Yes, and actually I'd like to mention that even when I tried to carry out the same test using different genetic labs, some showed no signal, like no DNA. So as I said, we still need to work on that, but I think it's very promising if everything is optimized, and yeah, we could be able to detect or actually analyze the small, because they are actually small fragments of DNA. They are not like an intact. And actually, I'd like also to mention that another research found out that the more we keep DNA in the culture, the more it's subjected to degradation. So it will give us negative results. So they are still working on deciding what's the best technique to collect the sample. But some people even say that we have to sample the DNA, or actually take the samples daily and put them all together and then send them to the genetic lab. You know,
that's interesting. I guess at some point, does it become just as labor intensive as doing biopsy if you keep doing that? Yeah, doing the roundabouts. Right, I think we are there for questions. Aya, that was absolutely fantastic. I appreciate all your time and effort. Not only the presentation itself, but the enthusiasm with which you gave it really came across there. So it's absolutely fantastic. So without further ado, this now leads me to introduce our second speaker. Abdullah Idrees is a senior clinical embryologist at the King Faisal Specialist Hospital and Research Center in Jeddah. He is also a PhD candidate at the University of Newcastle. So Abdullah, if I can just ask you to get your slides up, if that's okay. Just share your screen for us. And Aya, we might need you to unshare. That's fine. Excellent. So Abdullah is going to tell us about the clinical applications of confocal microscopy. And I guess a lot of this is around the research for his PhD as well. So Abdullah, over to you.
Yeah. Sorry, let me just. Yeah. Do you hear me? Hello?
Yes, we can hear you. Yes. You're welcome to leave your camera on if you like, or if you want to leave it off, Abdullah, it's entirely up to you.
So, good afternoon in the UK time. First of all, I would like to thank Kent University, Professor Griffin, Dr. Giuseppe, IVF Company, Shadi and Mayyas, Tri-C Academy, Mr. Munder, for having me to give you an idea about some clinical applications of confocal microscopy in the reproduction field. So let's start and get to know confocal microscopy a bit closer. Confocal microscopy is a microscope that's broadly used to resolve the detailed structures of specific objects within the cell. Similar to wide-field fluorescence microscopy, various components of living and fixed cell or tissue sections can be specifically labeled using immunofluorescence, for example, and then visualized in high resolution. As a distinctive feature, confocal microscopy enables the creation of sharp images of the exact plane of focus without any disturbing fluorescence light from the background or other regions of specimen. Therefore, structures within the thicker objects can be visualized using confocal microscopy. Furthermore, by stacking several images from different optical planes, 3D structures can be analyzed. Confocal microscopy has been traveled through a long journey last few decades to reach what we have today. The basic concept of confocal microscopy was originally developed by Marvin Minsky in the mid-1950s and patented in 1957 when he was postdoctoral student at Harvard University. Minsky wanted to image neural networks in unstained preparation of brain tissue and was driven by the desire to image biological events as they occur in living systems. Following Minsky's work, David Egger and Mujmer Bertrand fabricated a multiple beam confocal microscope in the late 1960s that utilized a spinning disk. Continuing in this arena, Egger went to develop the first mechanically scanned confocal laser microscope and published the first recognizable image of cells in 1973. During the late 1970s and the late 1980s, advances in computer and laser technology coupled to new algorithms of digital manipulation of images led to growing interest in confocal microscopy. Dutch physicist Fred Frankhoff developed a scanning confocal microscope in 1979, while Colin Shepard contributed in the technique with a theory of image formation. After that, Tony Wilson, Brad Amos, and John White, during the late 1980s, demonstrated the utility of confocal imaging in the examination of fluorescence biological specimens. The first commercial instruments appeared in 1987. During the 1990s, advances in optics and electronics afforded more stable, powerful laser, and other features have been developed. Modern confocal microscope can be considered as a completely integrated electronic system, where the optical microscope plays a central role in a configuration that consists of one or more electronic detectors, a computer for image display, processing, output, and storage, and several laser systems combined with wavelength selection devices and beam scanning assembly. Confocal microscopy offers several advantages over conventional wide-field optical microscopy, including the ability to control depth of field, eliminating or reduction of background information away from the focal plane that lead to image degradation, and the capability to collect the serial optical section from the thick specimens. Similar to the wide-field microscope, the confocal microscope uses fluorescence optics. Instead of eliminating the whole sample at once, laser light is focused onto defined spot at the specific depth within the specimen. This leads to emission of fluorescence light at exactly this point. A pinhole inside the optical pathway cuts off signals that are out of focus, thus allowing only the fluorescent signals from the illuminating spots to enter the light detector. By scanning the specimen in the raster pattern, images of one single optical plane are created. 3D objects can be visualized by scanning several optical planes and scanning them using a suitable microscopy software, or what we call it Z-stack. It is also possible to analyze multiple colors of immunofluorescence staining using a state-of-art confocal microscope that includes several laser and emission excitation filters. The confocal principle in epifluorescence laser scanning microscopy is dramatically presented in this figure. Coherent light emitted by laser system or excitation source passes through the pinhole that's situated in a conjugated plane or confocal, with scanning point of the specimen and a second pinhole positioned in front of the detector or a photomultiplier tube. As the laser reflected by the dichromatic mirror and scanned across the specimen is in defined focal plane, secondary fluorescence emitted from the point of specimen in the same focal plane pass back through the dichromatic mirror and are focused as a confocal point in the detector pinhole. However, there are many applications in the clinical field for using the confocal microscopy. I will give you some examples from my PhD project in reproduction. Reactive oxygen species are chemically reactive molecules that contain oxygen and hydrogen peroxidase molecules. They naturally presented in the body, and their effects are normally balanced by presence of the antioxidants. Oxidative stress occurs when ROS greatly outnumber of antioxidants. So excessive of ROS can cause damage of cell DNA. For example, a damage for DNA of oocyte, sperm, or embryo. These pictures were taken by confocal microscopy at the bioimaging unit in Newcastle University in the UK as an example of its use in ROS measurement. The first image from the left shows a degradation DNA materials in a blue inside the oocyte, and the green fluorescence reflects the ROS levels within the oocyte. The middle image showed a fertilized oocyte with the male and female DNA materials, the two blue color inside the oocyte, and both polar bodies outside the oocyte, while the green fluorescence reflects the ROS level within the cell. Last image from the right shows the multiple sperm heads, the blue color within the oocytes, and again the green fluorescence reflects the ROS level within the oocytes. This work has been published in Fertility Conference last year in Edinburgh in the UK. As we know, after fertilization process, embryo goes through several developmental stages to reach the final stage before the implantation, which involves the formation of blastocyst. The blastocyst composes of tryptoderm, a single external layer of cell, and the inner cell mass, an internal cluster of cell. The inner cell mass will give the rise of fetus, while the tryptoderm will lead to formation of placenta. The following slides will show you a clinical application of confocal microscopy in cell allocation in the blastocyst in a 3D. This clip shows an expanded blastocyst in day 7 after fertilization. The outer green cells are the tryptoderm, while the inner blue cells are the inner cell mass. This clip shows hatching blastocyst in day 8 after fertilization. Again, the outer green cells are the tryptoderm and the inner blue cells are the inner cell mass. This work has been submitted already to the European Society of Human Reproduction and Embryology and will be published in the coming conference. Embryonic genome activation is one of the most important events taking place during mammalian pre-implantation developmental stages, which requires nuclear reprogramming. This nuclear reprogramming entails epigenetic modification. Among these modifications, trimethylation of histone 3 protein at 27th lysine residue is considered as epigenetic modification. It has been demonstrated that critical role of H3K27ME3 in silencing the developmental genes expression during the embryonic differentiation. The following slides will show you a clinical application of confocal microscopy in epigenetic examination in a 3D. This clip shows the two-cell stage in day 1 after fertilization. The blue channel shows the nucleus, while the green fluorescence that attached to the nucleus shows the level of targeted epigenetic marker. This clip shows the 4-cell stage in day 2 after fertilization. The blue channels shows the nucleus, while the green fluorescence that attached to the nucleus shows the level of targeted epigenetic marker. Finally, the take-home message is medicine discovery is on the verge of the next revolution by utilizing advanced microscopy to enable better prediction of lead candidates. The answer is no longer take more images, it is how do we get more information from the sample? Thank you for listening.
Excellent. Thank you very much, Abdullah. That's fantastic. Some beautiful images there. Not that the whole point is to take more images, of course, but what I will encourage everyone to do, because I don't see any questions in the chat, is to throw some questions at Abdullah. It'd be nice to hear from you all. I'll start off. What do you think are the major questions that we can answer using confocal microscopy that are not all that easy with regular microscopy? So what are the big questions in human embryology that we can answer with confocal?
Confocal microscopy give us a great opportunity to discover a new era in treatment, for example, epigenetics or genetics materials, as well as we goes down and see the whole structure in the 3D, as I show you some videos. So it will give us a great opportunity to find out how can we treat some genetic disorders.
So how would that work then? How would you detect and then treat genetic disorders with confocal microscopy that you couldn't do ordinarily? Can you talk us through an example of that?
So for an example, in my research, I'm trying to find out the effect of excessive of fatty acid, or what we call it non-esterified fatty acid, in pre-implantation and epigenetics as well. So I treat the media with high NIFA and see the difference between the control and the treatment group. After that, I would try to add some treatment to convert this bad effects, if there are any.
So okay, so it's treating the embryo. Okay, I get it. Yeah, excellent. Are there any more questions from anyone?
Professor, we have a for Abdullah. So if you want us to launch it, please.
We have a poll question. Okay, go for it. Please do.
Okay, here we go, guys. This is for a lecture for Abdullah. Okay, so we're gonna give you a few seconds. Come on, guys. Let's get the answers coming.
Okay, so the question is, what is the main difference between wide field and confocal microscopy? So I want to select one of the following: depth field, reduce background effects, 3D images, or all of the above. So is it depth field, reducing background effect, 3D images, or all the above? Can I ask you to vote now? We've got about 20% have voted so far. So I think I've got a feeling I know the answer to this, but let's see where we go. So we are up to 35% voted. So keep going, please, if you would. Just a few more minutes before we close the poll. I'll do a little countdown for closing the poll, if that's okay. So let's get us up to 50%, and then I'll do a little bit of countdown after that. So keep voting, if you would, people.
It will be fun, Professor. We will count with you.
There you go. Right, keep voting, people. Right, we will close the poll in 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. The poll is closing. Fantastic. So I have to say, over 3/4, 76% said all of the above. Is that correct, Abdullah?
For sure, absolutely. They are brilliant.
Oh,
so proud of our students. Well done, everyone. Okie dokie. So, does anyone have any questions? Does any of our panel want to ask a question? Shadi or Aya, Mayyas or Giuseppe?
I just wanted to check. There is a few questions, but I'm not sure if it's for. There is a question. It says, "Who wants to be a billionaire?" Style.
Yes, I think that was just a comment on my hosting of the webinar. I wouldn't worry about that. Okay.
Abdullah, you are a billionaire, Abdullah. Okay. So
I wanted to ask a question about super resolution microscopy. Is this a technology that's available to you in Newcastle, or do you see a clinical use for it, or is it just too new, not out there yet?
Abdullah, you may be muted, Abdullah.
Yeah, sorry, we can't hear you.
We can't hear you, Abdullah. Do you want to?
I think he's scared, Abdullah. He wants to run.
We can't hear you, Abdullah.
Mute and unmute yourself, Abdullah. Maybe this is what helps.
And so we can see if we can clear the line.
Yes, we have a special unit, bioimaging unit at the university.
Oh, we do have a bit of a sound problem there, unfortunately. I think we've,
Sure.
I think your Wi-Fi might be just going in and out a little bit too much there, mate. Right. Okay. So,
Professor, do you want us to go through the PGT and the IVF course until we get connection with Abdullah?
Yes. Well, we don't have any more questions, so why don't I hand over to you then, Shadi, to talk us through that?
Okay,
perfect.
Yeah, we've lost you at the moment, Abdullah, so we'll just see if your Wi-Fi can back a little bit.
Now? Hello?
No, it's not great at the moment, mate. We'll give it a few minutes. Shadi, are you going to go through this with us?
Yes. Yes. Hello. Hi, everybody, and welcome again to the University of Kent weekly webinar. And also, we are very proud that we want to announce the genetic and the PGT training course that has been designed and certified by the University of Kent from the United Kingdom. And IVF Company was proudly brought together with the University of Kent to the Middle East and Africa and India with our partner Tri-C Academy that hosting in Dubai. So I just want to tell you a little bit about this amazing course that we have at the University of Kent. And I just want to dive into the course structure straight away. It's basically consists of 9 weeks. 8 weeks is connected with the University of Kent online. And 1 week we will do the training hands-on at our training center, Tri-C Academy. So I'm just gonna tell you how we're gonna start these courses on the 7th of March, like maybe a week from now. Basically, every course that we do, and this is the 4th intake, we're so proud about that. We have a live webinar with the University of Kent, Professor Darren and Dr. Giuseppe welcome the student at the University of Kent, and then we have lectures. It's been recorded through the faculty at Kent, and it's available 24/7 in our database and host at Tri-C Academy. So we will talk a little bit about Love My Genome film for Professor Darren, a little bit about reproductive genetics, fluorescence microscopy, and how to maintain your lab book, how to be a successful scientist. And also, we do have independent study and reading list that it's come from the University of Kent, and the student can access this for free. And then every week, we meet the student every Monday. We go through Q&A just to make sure they made the maximum out of that week. And then we go to week 2. We tell them a little bit about the PGT, reproduction and IVF. Also, we have a famous lectures by Professor Darren, which is the male plate disappearing, and the pre-implantation genetic diagnosis lecture as well is so interesting, and a lecture about how the blastomere are talking to each other, and then independent study again, and then we meet them on the 22nd of March on Monday to go through the Q&A with them to make sure that they did learn as much as they can in that week. Week 3 on the 21st of March starts. We talk a little bit about screening of chromosomal translocation in animal, PGT in animal, and we go into the embryo biopsy and micromanipulation. Also replacing lecture and practical classes with online alternative. We do independent study. We make sure that the student read as much as they can, and we meet them again on the 29th of March to go through a live tutorial with Kent University. And then in week 4, we go into a little bit on the biopsy blastomer tubing, moving biopsied embryos, a little bit about the master degree that we have at Kent University in reproductive medicine. We talk a little bit about the embryo grading for biopsy and vitrification, and the famous lecture, the dinosaur DNA. I really advise everybody should see this lecture. It's amazing. We do independent study again and live webinar with Kent University on the 5th of April. And this is the 4th week finished, and after that we go into practical videos that we recorded in the lab to help the student have insight what's happening inside the IVF laboratory and prepare them for the clinical practice, hopefully when they get into the clinical field. So we tell them about week 5, we're starting on the 4th of April, about aseptic technique, media preparation, culture condition for PGT, equipment that we use for the PGT, quality control protocols. We do live tutorial again in the end of the week. And then week 6, we go into day 3 biopsy. I know it's a bit old, but the students need to know all the technique is available. We tell them about the grading, the protocol for blastomer tubing, and also we tell them the old technique about fish fixation and slide preparation as well. They need to know that as scientists. We do their live tutorial, and then we move to week 7 and go into the day 5 biopsy, how we grade, how we what is the protocol, how we tube the the tracheotoderm for the tubing. And video 4, we go into a little bit CGH and NGS, the techniques that we are using for analysis. And then the last week, we give them a little bit introduction about the DNA extraction, DNA amplification, neck translation, and fish slide staining. And then we will end week 8 the online training. We will, of course, there will be assessment and presentation and exam at the end of week 8, and then they get their certificate. And once the COVID is finished and the vaccine rollout is done in Dubai, we will bring them for the exciting practical session, which is based on the animal gametes, and we will give them a little bit about the laboratory technique and good practice in our training center, Tri-C Academy, a little bit about media preparation, theory and practice, coffee break, microscopy training, and then give them a little bit about mini prep DNA from the agar plate and take concentration, purity reading, coffee again. I think people love coffee in Dubai. Set up amplification for the isolated DNA. We do a little bit of neck translation for the amplified DNA, and then we will do first session for the fish setup and the second session for the fish and microscopy. And then we do the karyotyping practical and karyo lab from Kent University by Dr. Giuseppe. And then the last day we will talk about embryo biopsy, but also we bring the student in group and we let them hands-on do embryo biopsy so they know how it's look like when they go in the IVF lab. And then we do biopsy vitrification, final assessment, and concluding remarks. And also we take them for 1 day to our partner Dar Al Hikma laboratory in Sharjah next to Dubai, so they can see the diagnostic lab, how it's been done, so they can get a bit of knowledge about what's happening. And all our assessment structure is coming from the master degree at the University of Kent. Thank you so much for listening, and I'm gonna give the screen to Mayyas, our faculty. She is a PhD student also in reproductive genetics, and I will leave it to you, Mayyas. Thank you.
Thank you, Shadi.
So yeah.
Okay. So can you see my screen now?
Yes.
Okay. So now we will go through the second course that is designed and certified by the University of Kent, which is the IVF course. This is also a 9-week course that has, sorry, I think that I need to go into a full screen. Okay, so this course is also made of 9 weeks. First 4 weeks are theory. The second 4 weeks are practicals, online videos, and the last week will be the practical hands-on week that we will do in our training center, Tri-C in Dubai. As an overview for this course, the first four weeks will be a theory presentation that brought to you by the University of Kent. Each week will have an independent study and a live tutorial at the end of each week. So as a quick overview, you will have a few presentations about reproduction and IVF, male reproduction, female reproduction, how to write a patient-facing leaflet, and how to talk to patients. Also, on the next week, you will have introduction to animal IVF, a short documentary about making babies, basics of reproduction. We will also teach you about sperm DNA damage. Professor Darren will explain to you about the Masters in Reproductive Medicine at the University. You will see presentation, lecture, and practical classes with online alternatives. So this is also brought to you by Professor Darren. Third will be about laboratory techniques and good practice in IVF lab, andrology theory, semen analysis practical lecture, about propagation and how to maintain your lab book in a lab, about meiosis in the oocyte, about embryo morphological grading assessment, and the last week will be about blastocyst, about aspects of time-lapse imaging, microinflation, ICSI, cryopreservation theory. For the second part, which is the practical videos that is recorded in the lab, you will see videos about aseptic techniques in the IVF, how to keep aseptic techniques in the lab, about how to prepare media, different culture conditions, equipment, the quality control and protocols, how we keep a good quality control and how to follow protocols in the lab. About semen collection also, you will learn how to do semen analysis and sperm functioning test, how to prepare semen, different methods that we use for each treatment cycle, how to do sperm freezing and thawing in the lab, about epididymal and specular sperm and retrograde ejaculation. Then you will watch videos about disposables and tools used in the IVF lab, how to grade oocytes, insemination procedure, and the last week will be about fertilization check, how we know that oocytes are fertilized normally, about embryo culture, how to grade our embryos, and then how to use the best embryos for transfer, and the last one will be about embryo vitrification. Last week, which is the hands-on practical that we hope that we will be able to meet you all soon in our training center after the COVID pandemic is over. So we will go with you through many practical hand-on sessions like animal in vitro production methods and tips, andrology practicals, how to do basic semen analysis, sperm preparation for ART procedures, how to do media preparation for clinical practice, then how to do oocyte retrieval, how to do in vitro maturation, how to use the microscope to grade and handle the gametes. Then we will have a practical about animal IVM, then how to move zygotes and how to score your embryos in the lab. You will have a hands-on training on cryopreservation. Then the best part, I think, which is ICSI. Everybody is interested in learning how to do ICSI. So each student will have the chance to do a practical hands-on on injecting real animal oocytes. Then we will do also some science communication leaflets preparation sessions. and then we will go to the final assessment and concluding remarks. So this is in general was the IVF course that is brought to you by the University of Kent. Thank you, everyone.
Excellent. Thank you, Mayyas. Again, if there's any questions, you can put them in the chat box. That's absolutely fine. We would very much like you to be joining us to do these short courses. We've got some fantastic feedback from the people that are doing it. As well, you can imagine, we went from hoping to offer them completely hands-on to following COVID almost a year ago now, to having to go online. But we think we now, as a result, have a better system. So we allow you to go online for a shorter amount of time, for about 9 weeks, and then we'll bring you into what we hope will be our new facility very soon to do some hands-on work. Shadi.
Professor, we have the poll for the 2 courses. If you don't mind, I launched them, please.
Go ahead and do it, please. Yeah.
Thank you. So we're gonna start with the PGT, my one. Thank you.
Okay, so would you be interested at the University of Kent PGT and genetic courses from the 7th of March 2021? Very easy choice. Yes or no? Oh, and look at this. What a wonderful audience we have. We have got all the way. We've got 31%, 32% voting. Keep voting, please, if you would. We'd love you to join us on our courses. Like we say, we find them very, very rewarding to teach. We've got a couple of our graduates have actually spoken today. So very, very good advert for that. And you can see what a wonderful job that they did for you today. So we're going to close the poll very soon. We've got nearly half of you have voted, and really lovely to see. So we'll close the poll in 5, 4, 3, 2, 1. Thank you very much. And delighted to say that over 80% of you said yes. So we will be holding you to it. Please do come and join us and do these. Next week, we have The Future of IVF by Dr. Syed Rabah, who is a Canadian certified scientific director, founder of the National Scientific Director of Nahal Fertility Program, Toronto, Canada. So we are going over to North America again, just to give you an idea of how international we are. So with that, I will ask Shadi to just say a few words.
Yes. Sorry guys, but I need to give you one more poll before Professor Darren close and give you a bit of interactive. One more last one for Mayyas, if you allow me, Professor Darren, before you close. For the
okay, go for it, yeah.
Thank you.
Yeah.
Here we go. Thank you.
Oh, so this is the same question, but this time, would you be interested in the University of Kent IVF course? So the last one was for PGT. This one is for the IVF course. So again, we're going to hold you to this. Yeah, put your bank details in the chat. I'm joking. So yes, we're voting. We're already up to 40% already. Thank you very much. And it looks like we're going to get some very, very similar answers again. So we are up to 47, 48, 49, 50% have voted. Thank you very much. So we'll close the poll in 5, 4, 3, 2, 1. Close the poll if you would. Thank you. And I'm very pleased to say we've got an 80% yes. So 4 out of 5 there. Okay, go ahead, Shadi.
And it's recorded, Professor. Just anybody want to ask for the video, it will be in our YouTube channel in 2 to 3 days. And Melissa, our operational manager at IVF Company, she will make sure that it's available so everyone can catch up to see Aya from Egypt and Abdullah from Jeddah and Newcastle in 2 to 3 days.
Excellent. And who wouldn't want to see them? They're both fantastic speakers. Does anyone else have anything else to add? Mayyas, do you want to add anything?
There's an extra question here that I think we have missed. One of the listeners is asking, will this course, I'm assuming both PGT and IVF, will those be offered again later this year? So if they can't
Yes.
make it for the 7th of March, will this be offered again? Will this be offered during the summer? And what are
Yes,
the plans?
We hope to run them throughout the year, and then as soon as we can open up our lab facilities, then we'll be inviting you over there as well. So yes, we will run them again, but obviously very much like you to join us in March if you possibly can. Anything else to add, Giuseppe?
No, well, I think we had two fantastic speakers today. I feel like I've learned something, so it was a pleasure to see them. And yeah, it's all for me.
Mayyas, anything to add?
I think Dr. Giuseppe is excited to have his fourth intake for the exam. He likes to make the exam for the students.
Oh, come on. Aya said that the exam was brilliant, so it cannot be too bad.
Yeah, I enjoyed it so much.
There you go.
I might take the course once again to just solve the exam.
Oh, we'd love to have you back.
Okay. I just want to thank Mayyas. Do you want to say something, or I just want to thank? Yeah.
No, just I want to thank
the net for Mayyas today. The net is not good for Mayyas. I want to.
I'm afraid we can't hear you, Mayyas. You're frozen. So without further ado, we will close now. So thank you very much to Aya and to Abdullah for fantastic talks. A really good example of our graduates from the IVF and genetics short courses. It's great to have you on board. We're so proud of you, and we're looking forward to more of you to come and join. So without further ado, thank you very much for joining us, and we will see you all next week. Thank you.
Thank you.